Okay, let's start session 3. It's mouthful. Innovative tools for open life sciences. So I'm Norio Kobayashi at Infrastructure Research and Development Division at RIH, 情報統合本部 in our institute. So in this session, we invited two speakers. One is Toshiaki Katayama-san from DBCS, and the second one is Kinoshita-san from Soka University. So first of all, let me introduce the first speaker, Toshiaki Katayama-san. So he's a project professor at the Biodata Science Initiative, BSI, and the Database Center for Life Science, DBCLS. So his research includes developing tools in various DBC projects that accelerate further integration and improve the interoperability of databases and services in life sciences. Furthermore, he has been organizing the annual hackathon events called International and Domestic Bio Hackathons, providing a place for discussion and joint development with various researchers. So the title of his talk is Services Developed for Integrated Fair Databases in Life Sciences. Fair is a very, very important property to integrate data sets and collaboration. Okay, please start. Thank you.
Thank you for kind introduction. First of all, let me apologize. I caught cold last week in San Francisco, and my throat, and both of my throat and my thoughts are not perfect, but let me do my best. Okay, so Database Center for Life Science is a national institute which integrates as many as possible life science databases. And to do that, we focused on the standardization and the interoperability. And to improve that among the worldwide database developers, we had been organizing annual biohackathons. And FAIR principles are also invented in the biohackathons in 2015, I believe. So the Semantic Web is the best implementation of the FAIR principles. And now we are asked where and how we can utilize those integrated database. So one of the applications might be a large language model. So how to utilize those heterogeneous datasets through chat API. Or we can combine emerging genome graphs, how to annotate the structure variations, or maybe we can combine the automated research workflows which machines generate the scientific data. So all of those are in our scope, but it's still a challenge for us. So why the data integration is important? So this is a quote from the review. And the author is accumulated the largest collection of the human genomes to evaluate all the variants. And so last decades, we have been sequencing as a lot of samples from human populations, and we made a lot of GWAS studies or fever studies. But the key point is we are still understand a very little limited number of the causability. So what is the mechanism? Why this variant causes this disease or phenotype? So as a scientist, we need to reveal the mysteries of those. And of course, it is intellectually interesting. So I would like to reveal all those interesting portion of the biology by utilizing heterogeneous data sets starting from variant genes, but with compound, glycans, diseases, drugs, or interactions. So, and 1 year and a half ago, I sequenced my own genome sequence out of my pocket money. And how to understand how to interpret my genome was a question. So this one was earwax type, and I'm dry, and obviously this allele suggested me I have a dry year works type. But how about other phenotypes like my characteristics or any other things? So to consult all those phenotypes of diseases, we need to visit a lot of databases from the variant database and the variant disease relation database and the protein database and protein annotations and protein data structure, protein 3D structure database. But still, it is very hard to interpret what the reason of the variant makes that phenotype. And at the same time, we require to deep biological knowledge to understand or interpret all those information written on the website. So it is still a very, very hard task to understand and integrate all those information. Okay, so first of all, to visit several databases related from the data you have, the identifier is also, of course, very important. So if you have a variant identifier, then you can go to gene to transcripts, and the transcript to proteins. So we can follow those biological objects by tracing the identifiers. And we are providing a Togo ID system where you can also contribute the set of identifier pairs to be converted. But at the same time, we found several limitations, like Glycan and protein has several semantically different connections, like this protein makes glycans, or this protein binds to glycans, or such kind of differences need to be distinguished. And also, only the identifier cannot reveal the, for example, if you have a variant location on a genomic scale, and if you want to see the variant where on the protein surface the variant is located, so not only variants, but the position need to be transferred. So that might be also true for spatial transcriptomics or any other new biology data. Okay, so to do that, everything in the database should be represented in a same standardized way. And we started to use the knowledge graph, or also known as RDF, as a basic data structure for over 10 years. So the data structure itself is very simple. All the database data are represented in a subject and a predicate and objects, those triples. And when those are accumulated and those identifiers are same, then the RDF graph will be naturally constructed in the database. And you can make a subgraph query by using a standardized Sparkle query language. But to organize the people utilize the same ontology or same identifiers among the databases, that is a human task. So we started a biohackathon meeting in 2008, and we discussed heavily about the data models, what kind of terminology, vocabulary need to be standardized, and what kind of identifiers or identifier schema we will follow, and how to open those data. So those community is, I believe it is great. And we made a lot of progress to combine all those biologically heterogeneous data sets. But still, there are several issues like new data sets, or we are not dealing with images or analyzing biologies yet. So we still need to extend our integration work. And we have been organizing 14 times of the biohackathon in Japan, but the culture is spreading around the world. In 2018, the European version of the biohackathon started, and some other hackathons are continuing with us. So every year we have been accumulating about 100 participants, and 30% of those are from overseas. And this is a one-week event, and we have entire 5 days for developing and discussing on the specific topics. So we usually have 20 to 30 subject groups, but you can define the group and you can resolve those issues in a group, or you can switch the group. So it is very, I believe it is very productive event, and you are most welcome to attend again. I saw some of those guys already participated. And one more thing we can note here is a bioarchive. So we tried to summarize what we did in 1 week in the past, and it took 2 years, so it is useless. 1 year event requires 2 years to summarize, so we ended up to make one great paper. And instead, we started our own preprint server, bioarchive, because bioarchive doesn't accept those review papers, so we did it our own one. Okay, anyways, we established an international community, and we developed a lot of standardized ontologies or mechanisms or softwares. Yep, basically, it is very hard to describe by words, but there are a lot of emerging new ideas and collaborations during a week. And finally, we have converted those very, very major bioinformatics databases, starting from genome sequence annotations to proteins to compounds to biomedical ones, glycomes, proteomes. So all those data sets are already available at the RDF portal in the same standardized format. So we now host about 60 databases here. So, and it's freely available, so you can make a query. And the number of triples we accumulated is about several hundreds, billions. So the number sounds like very big, but because we converted the original database to the RDF data model, so each data set has their own data structure. For example, this is one of the human protein, which related to COVID-19. And S2 human has a function description here and a catalytic activity here. And this page actually is very, very long. And all the contents are already converted to RDF by their own. Actually, they are managing the database system internally by using RDF. And the HTML website is just a representation of those. And that means they have 300 million proteins in their database, and that exploded into 100 billions of tuples. But still, it is manageable, thankfully. And this one is a PDB example. So they have only 200,000 records, but as you may know, several years ago, AI made the prediction of the old possible protein structures for 200 million structures. So by using the fact stored in a biological database, we can now utilize AI or large language models, new methods to complement the rest of the world. Okay, so still, again, we need to understand the data model you used or developed for each database. So the uniprot record is very, very long, but the data model is back-to-back. So how to pick the information portion which you are going to use in your research is a key question. So far, we have been writing a data model and making a generation of the Sparkle query automatically. But ideally, I would just like to ask him, get me all the kinases which bind to this, or something like that. So we do not need to write any query anymore. But we are now in a marginal step, so some part requires human curation. Anyway, so the heterogeneous data sources are now translated into RDF knowledge graphs, and we have standardized Sparkle query language, And to utilize those huge complex data, we created several softwares, like a Sparkle proxy and RDF config and Sparklist and Grasp, which I will describe very briefly. And after, by using those tools, you can extract some portion of the integrated data, then you can visualize or you can analyze those things. So we need to close the circle to accumulate, to accelerate the data transformation of the database ecosystem or in bioinformatics. So the Sparkle proxy is a very simple application which protect your Sparkle endpoint. So make your Sparkle endpoint completely read-only, and if the query was already executed by someone else, then the cached result will be sent back, so the response will be very quick. And you can also manage several things. So if this database uses different data URI prefix with this one, then we have a middle layer to translate on the fly all those prefixes, so that so we are now adding several of those tiny modifications to upgrading the utility of this middleware. Okay, the Sparklist is another tool which makes very, very long Sparkle query into just a REST API. So if you construct super complex Sparkle query, and if you want to use that Sparkle query just rewriting the gene identifiers or something like that, then you can put your Sparkle query in the Sparklist, and you can parameterize the gene symbols or taxonomy identifier of the query, and you can take those as parameters, and you can just use this REST API to extract the same information. And RDF config is our own data modeling tool. So writing the database schema like this takes a lot of time. So writing this schema by hand, I don't want to do that. So of course, writing down the data models in a YAML file itself is hard work, but still, if you made a data model change in the original database, then you can just modify few lines of the YAML codes. Then schema figure can be automatically regenerated, and you can automatically generate Sparkle queries. So the RDF config is a tool to support all those transformations. And by using this, we can also provide a GraphQL query against the Sparkle query endpoint. To make it possible, we developed a Grasp middleware. So this encapsulates Sparkle endpoint, and you can make a GraphQL query. It is very easy. Looks like a JSON, and you will get back JSON. So internally, it executes Sparkle query, but the GraphQL query is nicely interpreted, and resulting the set of the results are transformed into GraphQL compliant result in JSON format. So those tools might be useful if you are relying on graph data. Okay, so we to test the availability usability of the RDF graph database in a semi-production database, like a genome database. So we created a Togo genome database in 2013, which covers all the 20,000 species, all the gene annotations, and also environments or phenotypes. Those heterogeneous data are integrated in that. And by taking the human portion of this database, we started a Togoba database, which is a collection of the Japanese genomic variants. And now we have been integrated those clinical interpretation to publications of each variant or each gene. And as you will see, here is a variant located on the surface of this protein. So all those informations are nicely integrated. But still, we need to integrate more to make interpretation easier. Okay, so each portion of the database is reusable, so you can copy and paste each portion of the graphics in your database. So we are using the Togo standard and meta standard on most of our databases. So for example, if you would like to reuse the green bar notation for the variant dataset, then you can just write this code in your web page, then you will get this representation. So our focus is to decompose all those all existing database, not only in terms of the information level, triples, but also in the visualization components. So if everything was modular, our rebuilding new database process might be much, much easier. And another thing I have been involved in for years is graph genomes. And I was so excited to see this phrase, like a graph-based human reference genome is a game changer. It was 10 years ago, but people are still using linear reference sequences. And now, so 2 years ago to now, a lot of genome graph publications research are published. And one of the interest of us, because we are database people, so how to represent very, very complex structure variations. So this is a report made by Nomad Group, and here is manually curated very complex types of the human structure variations, and how to code this in the database had been challenge. And still a challenge. And we tried to understand this VCF and which number corresponds to which genomic locations and what is the orientation of those exons. So it is very, very difficult to understand. And, so I think the genome graph is nicely, naturally fit to capture from this region to this region have a this path or this path. So and this looks like a CNV with inversion or something like that. So why just don't we use genome graphs for representing structure variations? And to push that forward, we have been working together with Japanese colleagues called BISC community, Variant Information Standardization Curriculum. And now all the Japanese variant databases like Togoba and MGen and JBA and some other guys are involved in this. So far, the structure variations recorded in the genome database is very, very simple, just long or something like that. So, but we will get submissions of the very complex structural variations in coming years, so we need to be prepared. So we prepared a genome variation ontology for that. Of course, it is extensible. And how to capture this in a compact way in a database is still we are discussing about that. Okay, so maybe time is up. Okay, so the last part is Togo DX. So to show the integrated data for everyone, we have been accumulating all those informations, and some of those biological concepts are linked together, and each concept has different properties, specific properties like a tissue-specific expression or protein functions or 3D structures. So by using those information, you can narrow down or point where which and where in the database you should be looking at to extract more relevant information. So like a exploratory data analysis, EDA, you can make a query like genes expressed in the lung and localized on the cell surface and a 3D structure in PDB, then those proteins might be a target of the COVID cure, so drug development. So by combining all those informations together, you can make a hypothesis on the integrated database. Of course, the work is still in progress, so on the user interface, I think this is very beautiful, but I'm getting old, so it is very hard to read. So maybe another layer of the interface will be required. So that is why I also created this interface. So in the screen, on the screen, this is showing a human organ, and the organ follows your move. And gene expression of the set of genes are visualized on the fly, and each protein's description is shown on the second screen, and you can see the protein structure and the description there. So how to show and how to utilize the integrated huge amount of data is still a question, but we are making progress. So that's all. Thank you.
Thank you very much. Thank you very much for comprehensive introduction of tools and services of DB series, the life science data ecosystem. Any questions? Do we have time for 2 or 3 questions? So you have already developed the ecosystem for RDF data set in life sciences. But we also interesting in data management. I mean, we need to handle both metadata and data generated by two instruments and so on. Yeah. So they have a strategy to manage such life science data with RDF data.
Thank you. I think it's a very good question. And so metadata and data have a very much now. So it is very difficult to distinguish each other. And we in DBCLS having dealing with secondary databases like knowledges, but by collaboration with the DDBJ or some other institutions, we are now starting to explore the possibility how to make a repository from scratch. And to do that, of course, what is best is the machines used in the experiment output metadata as much as possible. And what metadata will be important in the future is not predicted completely, but still the machine types and lot, dates, temperature, humidity, all those things need to be recorded. And if we ask the data submitters to do that, no one does that actually. So those should be filled in by machines automatically. And my colleague in DBCLS is doing a research on the biosamples. Biosample have 4,000, 4,000万だから 40 million records. And each record has a really weird key and value pairs. So usually we would like primer as a key and a sequence in the value. But the primer sequence itself is located in the key. So if you allow the data submitters to use the key value pairs in any way, then it is very hard to constrain the standard. So what we need is identifiers and ontologies to cover as much as possible, but at the same time, it is not always comprehensive. So yeah, it is a problem of chicken or egg.
Much.
So, so my question is regarding the evolution of gene and transcript understanding, and how you know even something as curated as GenCode is still changing quite a lot, and when that propagates into something like your RDF networks and stuff, that I can imagine that can create lots of problems and things. I just want you to comment on that if you could, please.
Yeah, thank you very much. So at least what we can do is update the data regularly, of course. And we also observed the pathogenic variant in Climber became benign a few months later. So those changes often occur. So we need to trace identifiers and versionings as well. And by relying on which data sets of what timestamp made your research to conclude in this way. So to be honest, it is very, very hard to capture everything, snapshot of everything. But yeah, as a database provider, we will continue to provide some method to point to the right version of the data and make it referenceable.
So, for like ID migrations and
Yeah.
you've developed automation pipelines, or is there still a manual curation process where you need to have people double check things?
Yeah, so in INSDC collaboration, like GenBank and the DDBJ, they have their own versioning, controlling, and trace. And our database, like a Togoba, we internally have it, but not so much heavily used. Because our database updates is once or twice a year, and most of those are still okay. So, but the identifier merge or separate, yeah, I understand that can happen a lot in the future. And especially for structural variations, it is very difficult to identify those two structural variants are identical or not. So yeah, identifiers and how to point that is a endless problem.
Any other questions, comments? So would you like to introduce your next biohackathon event here?
Oh, yes.
And Togo-san?
The dates and the place are not confirmed, but we are trying to finalize it July 5 or something like that. July 5 to 1 week in Fukui Prefecture. So I need to go there to see if everything is okay. But potentially, we will organize 1 week hackathon in July. So if you are interested, please visit biohackathon.org, and we will make an announcement on Slack mailing list. Thank you.
Okay, thank you very much. So let's thank the speaker. Thank you very much, Katayama-san. Okay, let me introduce our second speaker, Kiyoko Kinoshita-san. So she is the Deputy Director of the Glycan and Life Systems Integration Center at Soka University. Her research includes glycom informatics, the study of carbohydrate sugar chains using computer-based technologies such as data mining. She has been developing semantic web-based databases and is currently working on developing a human glycom knowledge base called TOSA. So today she will introduce the GlyCosmos portal, which comprehensively integrates omics data such as genome, proteomics, diseases, and pathways. The title of her talk is Using Inference on Semantic Web Data to Enrich the Data in GlyCosmos. Please.
Thank you. Thank you for the invitation to come here. It's been really interesting. Right, I think it's not based on all the talks from today, it seems like everyone knows about the semantic web, so I may not need to go through all of this. It's also known as linked open data. There are whole, not just the life sciences, but there's, geography and government and all sorts of other types of domains that are all linked together in the LOD. The life sciences cloud is also quite heavily, connected. But just one thing, I just happened to come across this paper in 2021, and I was just looking for some examples of semantic web, and there was this meta-analysis of the life sciences cloud, and I was looking through this, and lo and behold, this was my database. I was so happy to see that. So this GlyCan is the Glycan structure identifier for all glycans. So if you go to glycan.org, this is a repository, you can register any kind of glycan structure and get an ID. So this ID is what links all these other Glycan-related databases together, and it's what we've been developing at Soka University and with the JST funding since 2017. So but not just connecting data, but we want to make use of that data, it's a knowledge graph. So we're looking into how we can do some inferencing and creating, finding new insights. And so the inferencing requires ontologies and rules. Basically, having the idea of what the modeling of the data model, and then defining, and applying rules to predict new knowledge. So we've been doing a little bit of that on our GlyCosmos data. So I'll give a few examples. But before that, I'd like to just briefly give a kind of a primer on glycobiology, just in case. So on the cell surface, on in mammalian organisms, any cell that you go and you find, the cell surface is covered by glycans and glycoproteins and glycolipids. So this is the variety, actually it's an old picture, but this is a variety of all the different glycans that you can find on the cell surface. They are connected to lipids. They're on proteins, the long proteins called proteoglycans. There's GPI anchors where there's lipid this thing. So GPI anchors and very complicated structures that cover, every cell in our body. They're also very heavily, connected in the extracellular matrix, interacting with collagen, fibronectin, integrin, etc. This is even a picture of a cell, an endothelial cell, and all the glycocalyx can be seen as like a hairy object. This is all the glycans on our cells. And so every cell in our body is covered by this. But everyone ignores them because it's just too complicated. So we've been trying to make it simpler, more accessible to access this data. And so there have been some standards that have been developed to represent first glycans using these symbols. This is called the symbol nomenclature for glycans, SNFG. So any publication nowadays will be using these symbols. So this is the glycoinformatics has been kind of really organizing itself in the last decade. So, like I said, glycans occur everywhere. They're on proteins and lipids, and some even say that they've seen them on RNA. So glycoRNA have also been identified, and they serve a lot of different functions as receptors in cell signaling, in development, you name it. So the major classes of glycans include N-glycans, O-glycans, glycolipids, the GPI anchors, and glycosaminoglycans, or GAGs. And during the biosynthetic process is really complicated because it's not like RNA, DNA, RNA, protein, the central dogma. It's a translational, it's not copy and paste. You cannot just predict a glycan. You have to know the different enzymes that are involved in adding each monosaccharide one by one onto the glycan, which then goes to the Golgi, and all the localization inside the Golgi affects what the glycan looks like at the end. And by the time it gets to the cell surface, you get a whole lot of plethora of glycans on the cell surface. And based on those different enzymes, you get different, congenital disorders like glycosylation. If you have diseases and these are knocked out, you'll find that the glycans that are changed then have caused major diseases, and they're called the CDGs. So, and they're also involved in various signaling, and they're recognized by these other proteins called lectins. So on the host cell, the lectins, these proteins will bind to the glycans on the cell surface and cause various functions. So in the GlyCosmos portal, we've been trying to organize all this information into a single website so that anyone can come and learn about glycans. There are 4 major sections. The submissions is where we have the repositories. So I mentioned GlyTucan earlier, but a lot of mass spectrometry is used to identify glycans. So we have mass spectrometry repositories. We're also developing a glycoconjugate repository for glycopeptides and glycoproteins. And then we just recently released in December a lectin microarray data repository. And then the actual integrated data is in this data resources section where we kind of divide it by data type. So we have glycomes, glycans, glycoconjugate glycogenes, lectins, diseases, pathways, and organisms, and various tools as well as the standards that we base everything on. So in there's a whole, there's so many different data. So the pathways kind of an exemplary view of how we've connected all this information using the semantic web and RDF in the background. So we've worked hard to try to make a user-friendly interface so that people can come and learn about glycans more easily. So this is an example of the pathways that we have. We extract the RDF data from the Reactome and we've gotten all the Reactome pathways that have any glycoprotein in it. And so this is an example of the keratin sulfate degradation pathway. Keratin sulfate is a GAG, a glycosamine glycan. So, for example, you can click on this one. This is the glycan. So there's a glytucan ID assigned to this glycan structure. If you click on the enzyme, you see the enzyme that works on it, that produced this glycan structure. The green circles are proteins, and these are actually a complex of these different types of these different keratin sulfate core proteins, and these icons indicate that this is a glycoprotein. It's kind of hard to see. The glycoprotein, which also includes information about which site on the amino acid sequence is glycosylated, and these are the glycan IDs. These are all mass spectrometry based glycans, so these are mass glycan compositions, monosaccharide compositions, but all these different glycans have been identified at this position 464 on this protein, for example. And these are linked, of course, to the glycan structures. And then we have the lectins which bind to glycans. This is not actually the same lectin, but the lectin information would give you more information about what lectins or what glycans it recognizes and binds to. So this complicated relationship of glycans to proteins can be kind of seen here, where we have enzymes working on glycans, but also being glycosylated themselves, and then linking to other glycans, in this one view. And so hopefully it's user-friendly for people to learn and delve into glycan function. So like I said, we're trying to get everything into RDF. So we've developed some standard RDF ontologies. So the naming was kind of poor here, but this ontology is called GlycoRDF. So we're linking glycan structures with the citations, the biological source, and their evidence, the experimental evidence that was used to identify that glycan. And so we have identifier, or your eyes for all this information. Also, a glycoconjugate ontology was developed so that we can reference not just the glycan themselves, but the proteins that they are on. Where? Here. The reference protein, or if it's on a lipid, the lipid information, and we've defined ontologies to use for this. And more information. We've tried to reuse existing ontologies where possible, using Faldo and UniProt, and just trying to basic standardized formats as much as possible. In the tools section, this is where we've been collaborating with experimentalists to try to develop tools to understand glycosylation better. There's some drawing tools, but then we also have tools to visualize glycogen expression information onto glycan pathways, or this LM, like a glycome atlas tool that has glycan distributions in various tissues of human, mouse, and zebrafish. And some HPLC data of glycans, glycan structural alignments, simulation of glycan biosynthesis using ODE simulation, reaction kinetics information, and then mass spectrometry annotation software. Just as a quick example of using GlycoMaple, so we have, this is all handmade pathways of glycan-related pathways, so this glycosylation or glycobiosynthesis pathway. And so by uploading gene expression data or RNA-seq information, you can then see and visualize where the enzymes are more highly expressed or more lowly expressed. And this gives you an idea of what glycans you might find in your sample, because not everyone can do mass spectrometry. So it's a way to try to make it more accessible to understand what kind of glycans you might be expecting to find on in your sample. And we have different ways to display this information. As an example, that you can take, for example, the breast duct cancer gene expression data, from GEO. Just you have this table of the expression data for each gene, using the gene names. You can have 3 different samples here, and uploading it into GlycoMaple. You can specify which ranges to use for which, and what arrows you want, the strength of the different ranges of the arrows that you want to specify, and that way you can see where the highly expressed genes are. These are not found in the sample, so they remain red, but yeah. And then at the end, because through this, you can also do comparisons as well. So in this case, you can compare a DCIS sample versus healthy with, if less than half expression would be green. If it's more than double, then it becomes red. And so a quick analysis shows that, in this DCIS versus IDC is not much of a difference, down here, but versus healthy, you'll see that these Lewis structures are quite downregulated. So, and these Lewis structures are known to be signals or diagnostics for cancer. So it was nice to see that we can kind of recapitulate information that's been already known. So, how much time do I have? Okay. So in this current project in the GlyCosmos that we're receiving support for, because we tried to accumulate as much data as possible from existing data resources, but that was not enough. There's so many other information that's still in the publications, in literature, and has not been organized anywhere. And so our new project aimed to expand our repository collection. And so this latest release included this new one, LM repo, to be able to accept more information or more data from experimentalists. And so we developed all these we're developing repositories which can then be automatically or semi-automatically curated and to supplement our data resources section. And another thing is the semantic inferencing is also another thing that we've been trying to do for that. And these are all based on the Mirage guidelines, which I'll explain a little bit later, but these are standardized guidelines that have been proposed for experimentalists to use when uploading their data into these repositories. Right. And another thing, we've been working internationally as well. So there's a project in America funded by NIH called GlyCan, and there's another project in ExPacy in the SIB, so the Swiss Institute of Bioinformatics, called Glycostatic ExPacy, and then our GlyCosmos, we've come together and formed this GlySpace Alliance to ensure that we share our information. We provide trustable information, and if one site goes down, that we can cover for each other in case something happens. So it's a way to kind of ensure that our infrastructure is stable for the entire glycoscience community, and we meet regularly to make sure that we're communicating and stable. And this is the Mirage guidelines. It stands for minimum information required for a glycomics experiment. A lot of text. The aims are to develop a reporting guidelines and also to use it as a communication hub. These are the members. So we meet in Germany every year to discuss. And these are the guidelines that we currently have out. So we have sample preparation guidelines, which is kind of global to all these other experiments. Mass spectrometry analysis, Glycan microarray, liquid LC, NMR, CE, and lectin microarray. So we make sure that these are guidelines just for reporting in manuscripts. They're not related to any ontologies, and so then it's the bioinformatics people in this group, their responsibility to make sure that we can map this to ontologies, to standards, to make sure that this information gets stored and uploaded into repositories. So it's been kind of a cooperative thing that we've been doing. Right, okay, and so finally to semantic inferencing. So the data in GlyCosmos, we've accumulated was not really focused on taxonomy or on the organism, so we were trying to collect anything from any organism possible. But as a result of that, we have 24,000 taxonomic annotations, and it's like huge. So we created a table, let's see what we have. And when we looked at it, we're looking at the different glycogenes, the glycans, glycoproteins, lectins, and pathways. And looking at it more closely, we see that, for example, if we focus on one genus, this is all Aspergillus genus, we see, okay, there's glycans, okay, good, but okay, in this Aspergillus genus, there are 10 glycans, but Awamori has 14, and Aspergillus niger has 29. That doesn't make sense, right? So we're looking at, there should be more. It should include all of this. So we're thinking, well, I don't want to go through all of this hand by hand, one by one. We should be able to inference this. So thanks to Katayama-san, he had this taxonomic tree from NCBI, which we could use as a main graph, as an inference graph, and we just stuck it into our graph database. And as a result, very easy. So we just set up some rules to infer from that inference graph, and we made a query, again, for Aspergillus, to see what we would get. And we are able to then expand our list. This is hard to see. The result, sorry. We're successfully able to retrieve all glycans under the Aspergillus genus, including all the species. So this is a query for all of Aspergillus, but it included also the other species inside this genus. So we were able to, without having to change our query at all, we could automatically get what we were expecting as humans, logically. So we did not add any new data or anything. We just add that one species graph, and was able to do this. And so that really showed us the strength of the semantic web technologies. And so we've expanded this more inferencing tools into our interface, into our data. We have, I don't have any slides on that, but our future plans is also to continue doing that kind of similar thing, but now not on the taxonomy, but on the actual glycan data, which is also kind of complicated. So if you look here, these are all the same glycan, actually. But they all have different glytucan IDs. Why? Because they use different technologies, or there are different levels of ambiguity. So they all have the same mass. So the MS1, mass spectrometry, MS1 at one level would give you this kind of 5 hexoses, 3 hexanax, and 1 salic acid structure. But if we can identify the stereochemistry of these, then we can get 4 galactoses, 1 glucanac, 3 hexanax, and 1 salic acid. They're the same mass. This could also be this structure, which don't have any linkage information. We don't know what the linkage conformations are. Or at the most detailed level, it would be this structure. And so this relationship should be inferable, right? If we know one thing, we should be able to infer. For example, if we know that this one has some taxonomy annotation to it, then this should also have that same taxonomy annotation attached to it, although it doesn't at this time. So that kind of information can be now inferred once we get this ontology set up and installed. There's another concept called archetype. So we have these 4 glycans with different glytucan IDs. Don't they look the same? To the biologist, they're all the same. However, they're only different because of this reducing end, which is attached to the protein. So for a chemist, it's a different structure. We cannot have the same ID. So this is a beta structure, this is an unknown structure, this is actually, the ring is unknown, and this one is the archetype structure. Anyway, they're different, but they're the same. And so we define this thing called archetype, where you select one of these. This is the most encompassing structure for all of these. It's the representative structure for all of these. And we can map them to each other And so we can compute this information, and we're working on that right now. And we have a poster later that in the poster presentation to discuss this. So this was also another target for our semantic inferencing. All right. So yeah, GlyCosmos has been renewed. We're in the middle of our 5 years to develop new repositories for glycan pathways, actually for microbial glycans, for glycan microarray, and for lectins. And these repositories will then be semi-processed or semi-automatically processed to supplement the data in the data resources section. Semantic inferencing has enabled us to further supplement our data and organize our data better in a way that's logical. We're not adding fake information. It's based on logic, hopefully. And one thing I want to emphasize is that AI is so popular now, but we have to provide the high quality data to have high quality AI, prediction. So I think this data science, is a very important part of this, and I hope that you all agree. Oh, and then the Human Glycome Atlas project has just started in April 2023 under the large-scale academic frontier promotion project of MEXT. So the goal is to create this human glycan knowledge base. So there is no human reference glycome right now. We don't know what the reference glycome is for human. So this project aims to standardize that. And so there are different parts to it. The human glycan precise map. We also are planning on accumulating 20,000 cohort in the first 5 years and then 200,000 after that. And then also an atlas of the glycan biosynthesis process. So this is all going to be coordinated and stored into this knowledge base, TOSA. It's quite complicated because it's not just this part, which includes these different segments from Nagoya University and Gifu University and the National Institute for Natural Sciences, and all being stored in this knowledge base. Lots of different ontologies, including imaging information, is also going to be part of this as well. We also have developed the equipment and infrastructure for that, and based on this, expand on collaborative research, not just within Japan, but also out overseas. So this is a big 10-year project, which just started, but basing everything on standards, that will hopefully extend the central dogma, because we believe that we can't explain life just with central dogma. We have to include those glycans. All right, so thank you. These are acknowledgements. Lots of people have been involved in this project, lots of funding, and I appreciate all the support. Thank you.
This is also an ecosystem of glycoinformatics, including ontology, RDF, tools, software. Any questions?
So I'm also RDF people. So I believe one of the advantage of using RDF is we can use we can sharing the middleware or tools. So I believe to you and Katayama-san and Kiyoko-san closely collaborate. So, but how many middleware do you use developed by the DBCRS or something?
Middleware meaning.
Yeah. So for the GlyCosmos. So do you use some middleware developed in the DBCRS?
We have endpoints. We have APIs.
APIs.
Yeah.
So maybe so. I have a question for the Katayama-san. So using RDF or something. So can the domain, cross-domain sharing of the tool is how many shares tools are shared for the?
Yeah, we used a lot of his. We used Sparkle lists, and we used I think we used the Sparkle proxy as well. We used a lot of their technologies. Yeah.
I don't have exact numbers, and we are asked to increase the number of the users and our tools. But at least the Glyco community is great. They are natural born Semantic Web database community, and Yeah, so, and in a microbiome group, they are using Togo stanza and probably Sparklist as well. And for our area, variant databases are now going to we are sharing the data in RDF, so we will provide some visualization modules and probably query modules in common in Japanese variant databases. So it is gradually spreading. I hope everyone will start using that, but yeah, it takes time.
Thank you very much. And also, you are developing some global alliance of the GlyCosmos. So, how about the GlyCosmos community? So, in GlyCosmos community, you're sharing some tool or something.
Yes.
Uh-huh.
Yes.
So, different level from the RDF.
Yes. Yes. So user interface. Even for the repository section, so actually, 2 weeks ago, the GlySpace Alliance came to Japan, and we had a week-long meeting to discuss and share information. And so, yeah, they're also developing tools for example, the repositories. Like here, we could use more help, with just helping users upload their data, and based on ontologies. So they'd developed a tool called Table Maker, which includes a way to make it easy to map ontologies to their terms in a table file format or something like that. So, yeah, we're sharing tools and software and resources across the other alliance members.
So this is some case of the, so you do some sharing, across the domain and also so in the domain. So how do you think? So it is a typical case of the data interaction for in the biology or something.
No. I think we're lucky in glycoscience. We all we get along. I've heard about, I don't know about proteomics. There's maybe some issues in proteomics has kind of been slowly trying to figure things out. It's starting to finally get together with HPP and HUPO, and we're also actually working. We have a glyco working group in the HPP as well.
But I think to sharing tool is very important for the data integration. So how do you think?
Yes. Yeah, so we're working on going to conferences and talking with users, with actual biologists, and sharing our tools and databases, so that and getting feedback about its user friendliness. This is no good, or that's okay, but that's not so good. It's lots of feedback and communication is needed, especially repositories. It's difficult to get users to upload data into the repositories. That's the very big bottleneck. So we're trying to think of new ways to make it as easy as possible to upload metadata with their experiments. Just trying to figure out new ways, and using ChatGPT to make queries on the RDF data is another big topic right now. But it's more communication. I think is just the big key. And trust.
Thank you very much.
Any other questions? This is domain specification, so maybe wet researchers are interested in how to use the data and how to analyze the data. Any questions? So I'm a researcher of information science, so it's a dry question. So what kind of AI do you need to improve your knowledge graph currently?
Well, I think the inferencing, has been really useful, and
like LLM or large language?
We're looking into LLMs. I've seen DBCLS people developing specialized chat GPTs. So we're going to start looking into that and see if it's make it easier to query the data in GlyCosmos.
Okay, thank you. Any questions? Okay. It's already it's time. Okay, thank you very much.
Thank you.
Kiyoko-san, let's thank to the speaker. And this is the end of the session 3, Innovative Tools for Open Life Sciences. Let's thank to the two speakers again. Thank you. Thank you very much.